So a word bank would be like if you had a table there with like possible terms that you're going to plug in, right? So it's just from your head. You just have to remember them. Yeah. That makes sense? So it could go either way. Yeah, we could have the definition there, and then you just put the term in. That's where a word bank would come in handy, but you don't have one. Or it could go the other way. Or it could go the other way. We give you the term, and then you write out the definition. Yeah, so either way. Okay. All right. Okay. And then reminder on the quiz that's due tonight. Okay, so that if you haven't done that quiz yet, it's due by 11:59 p.m. Remember to use Chrome. Okay, use Chrome as your browser. Don't go in and out of it. Okay, just sit down when you know you have time. You know you have a good Wi-Fi connection. Don't try to do it on your phone when you're walking to somewhere. Okay, just make sure you're ready to go. Okay, all right. On the lab quiz, yeah. So the spelling has to be close enough so we can obviously tell what you're trying to say. Okay, that's the basic rule on that. Yeah. That's just it's a Canvas quiz. You do not need to use HonorLock for that quiz. It's open book, open note, and it is off of the notes. Okay, and so just to clarify that, I have a couple of questions over email about that. So the exams in this class are over the notes. Okay, all of that material for the most part is in the book too, but I don't pull material from the book that's not in the notes for the exams or for the lecture quizzes. Okay, so there's anything on Friday, the lecture quizzes or the exams, that's from the notes. Okay, the Master and AMP might be things that are from the book that I don't talk about in the notes, but the purpose of that is more to get you to read and get ready for class. Okay, all right. Any other questions or things you'd like me to clarify before we get going? Yeah. The lab quiz is written on paper. Yep. Mm-hmm. What else? Other questions? Okay, go ahead. Yeah, I mean, you have about 10 to 12 minutes to take it at the beginning of lab, but it tends to be plenty of time for everybody to take it. Yeah. Mm-hmm. Anything else? Okay. All right. All right, guys. So today we're going to, you got some more questions? Yeah, go ahead. Yeah, how many is it? It's probably, I think it's around 15 or something. Is that right? Yeah, don't worry about that. Yeah, yeah. Just study. Yeah. Yeah, just study. Yeah. Study hard. Go ahead. Yeah, the lecture quiz is over the first 2 sets of notes, not including this stuff we're going to go over today. Yep. They're typically always over 2. And when you open up the lecture quiz, you can look at it, you can click on it and see without actually starting it. And it right there in the description, it always tells you which 2 packets it's over. So you can always go to that to check, just so you know for the future. But yeah, it's over the first 2. Okay, go ahead. Are you talking about the big PDF of all the notes, or what do you mean the lecture stuff? Oh yeah, so when you go, I can show you right here. So when you go to these weekly overview pages, so see the little YouTube link there right next to the thing? That's where you can go. So every weekly overview page has that. Okay, guys, if you're looking at this, she asked where you can access the videos, the YouTube videos from class. They're right there. Yeah, that's annoying. Yeah, so right here, okay. I've noticed if you're looking at it on your phone, then you have to grab onto this and kind of scroll it over that little box. You can't see those, okay? But it's on all those weekly overview pages. You could also go to the syllabus, and it has the whole schedule for the whole semester in there, which is the same as those little sheets there. So the YouTube links will be there too. Okay, so either place will be fine. There's also a playlist on YouTube under my YouTube channel, which is just Bio 1030 Fall 21. So if you subscribe to that, you'll also get a notification every time I upload one of those. So however you want to do it. I'm not trying to get YouTube followers. Believe me. I'm not. Yeah, you might have to actually. Yeah. A couple of my videos went viral, but no, just kidding. Yeah. What else? Other questions? Okay. We good? All right. Okay. So, yeah, and you guys can tell me at any point if it's just easier to not have that up there, but it's probably best to still have it. But I know that's really annoying to have it flickering like that. So we'll make sure to get that fixed before next class on Wednesday. Okay, so today we're going to kind of transition over to still talking about the chemical side and the chemical level of anatomy into the cellular side a little bit too. So taking our next step up into that kind of little bit of cellular physiology here. Okay, so a very important definition here and very important part of anatomy and physiology is solutions, because your body is a bunch of different solutions. Okay, and so the definition of a solution is a homogeneous mixture of components, and a solution always has both a solvent, that's the liquid part of it, and multiple different solutes. Okay, those are the substances that are dissolved into the solvent. Okay, so if we got the solvent plus the solute, that's what equals our solution. Okay, so the homogeneous mixture is a key piece of that. So that means it's liquid enough for everything to move around and kind of even out. Right? So by homogeneous, that means if we just took a scoop of it from one part of your body and then a scoop of it from another part of your body, if that was all part of the same homogeneous solution, then the relative amount of each solute and a relative amount of solvent in each of those scoops would be about the same. Okay, and so that's what that means. And so obviously we talked about water last time and the fact that it's a universal solvent, so it dissolves all of these different types of particles, and so we have lots of different things dissolved in the solution of our blood, in the solution of our cells, and then the third space that we would talk about would be in the solution of our interstitial space, which is space that's not inside of cells, not in blood vessels, but everywhere else in the body. Okay. All right, and kind of somewhere between a solid and a solution is a colloid. Okay, and this is a heterogeneous mixture of substances, both the solute and solution. Okay, it's a little bit more jello-like. It doesn't necessarily all mix around perfectly and evenly. Okay, and so this is a little bit more like what our cell cytoplasm is like. It's not quite a perfect solution. It's not totally homogeneous. It's a little bit more on the colloid side. Okay, so many of those components inside of solutions are salts. Okay, and we talked about ionic bonds last time. Okay, so and so salts are typically bound together with ionic bonds. Okay, so the sodium chloride here, again, the sodium gave up an electron to chloride, and then they're oppositely charged, so then they stick together. Okay, that's the ionic bond. And so all ions are electrolytes, which means that they're substances that can conduct electricity. And we'll talk a lot more about that when we get to the nervous system and how we activate skeletal muscle. But for now, those will be important in terms of electrical signals and other things in the body. Okay, so properties of salts, they kind of crystallize when they're in their solid form, and they dissolve easily in water. Okay, and they dissolve because of the polarity of water, right? The positive side of the water will stick to the negatively charged ion, negative side of the water will stick to the positively charged ion, and it'll pull that stuff apart. Okay, any questions about any of that? Okay. All right, so let's look at water as a solvent here. And let's take a second, and you can kind of look at these three different solutions here, okay? And just talk with the people on your team by looking at what the substance is, kind of what the substance looks like when you look at it and look at the charges around it, and to answer these two questions here, okay? In which of these scenarios to the right would water mix well with the solutes? And then just kind of discuss what's going on here. Okay, take a second and talk with the people next to you about that.
Yes.
All right, so let's come back together. And so what do we got? What about A there? So sodium chloride to salt, would that one mix well? Yes or no? Yes, for sure, definitely, because it's an ionic substance, right? So just like we just talked about. What about carbon monoxide, which is a polar molecule? Will that one mix well? mix well? Yes, that one would too, for sure. Yeah, because just like water, the C and the O have the opposite charge, right? So the positive part over here on the carbon sticks to the negative part over here on the oxygen, and it can pull that apart. Okay, but then we got a non-polar molecule like methane. They don't mix, right? So you would see it divide in like that, and they would just be in two separate areas. Okay. All right. Any questions on that? Okay, so then let's transition into what this looks like inside of our cells, okay, which is the cytoplasm, okay? So the cytoplasm is the contents of the cell between the cell membrane and the nucleus. Okay, so what's inside the nucleus doesn't count as this. What's outside of the cell membrane doesn't count as this, but everything else in the middle. So the main components of it are the cytosol. So this is this kind of somewhere between a solution and a colloid type of fluid inside of our cells. Okay, there's lots of dissolved things like salts, glucose, gases, amino acids, proteins, ATP, lots of things in there just floating around. Okay, then we've also got organelles. Okay, organelles are those little structures inside of cells that carry out lots of different functions. And we'll talk about all those different ones coming up soon. Okay, and then we got inclusions depending on what type of cell it is. We might have unique things like, for example, skeletal muscle cells have something called a sarcoplasmic reticulum, which is similar to the endoplasmic reticulum, but a little bit different. It's there to store and release calcium. Okay, other cells don't have that. Okay, and then we've got the cytoskeleton. Cytoskeleton is kind of the structural part of the cell. Okay, gives it shape, allows it to move around sometimes, allows things to move around within it. Okay. All right, so the cytoskeleton is made up of 3 primary proteins. Okay, and these proteins are the actin filaments, the intermediate filaments, and the microtubules. Okay, so the actin filaments are made up of the protein actin, which is one we'll talk about later with skeletal muscle as well. And the functions of it are it gives the cell shape, forms its shape, it strengthens the cell, and also allows the cell to move in the cases that it's a cell that does move. Not all cells do, but some do. Okay, we've got the intermediate filaments, which are actually the most stable and most permanent. So there are big time supporters of cell structure and shape. Some of these other ones, like microtubules and actin filaments, they can get broken down and put back together constantly throughout the life of a cell. But the intermediate filaments are pretty much solidified in where they're at. They're giving the cell its shape. Okay, those microtubules are the biggest. They're hollow. Their function is to kind of create this strong framework for the cell, but also function as little tracks for things to travel up and down the cell. Okay, and that's what this next slide has a little video on it. So I'm going to switch over to the laptop so you can see that. Hopefully it's not too frustrating to try to watch it with this, but we'll see how it goes. But it is a cool little video to show how microtubules work. cool sound effects. Yeah, sure. So that's a microtubule that a little vesicle is traveling on. All right, so yeah, so a little bit of background on that. There's two main proteins that are involved in that they're called motor proteins called kinesin and dynin, and those are the little ones that you see literally walking along those structures. So maybe it's just the Apple TV, huh? Would you guys prefer if I just went from the laptop? I can't write things in that way, but must just be the Apple TV that's giving us trouble. I can switch over to that. I do have to change how I'm recording though real quick. Give me one sec. Yeah, you don't want to record in with that infinite screen going constantly. So let me just fix that real quick and we'll get going again. Okay, I think that's working. All right. There we go. Okay, so just a little bit more background on that is that those little vesicles you saw, those are things like it could be a packet full of proteins that need to get to the other end of that cell to help build that cell. One great example of this in our body is the way that our neurons work. Okay, so some neurons in our body can be up to 2 to 3 feet long. Okay, because the body of that neuron, so the cell body of the neuron, where the nucleus is, where all the structures are made, is in your spinal cord. But then it sends its axons out all the way out to the tip of your finger, out to the bottom of your toes. That's all the same cell still, right? And so all the proteins required to make that axon and all the enzymes around through that, all of those things need to be made in the cell body, but then carried down to the end of the axon, right? And so that's exactly what the microtubules are for, is to provide these little tracks for those things to walk on. And so really, like it showed there, I mean, it's just literally just tons of traffic, that vesicle traffic just back and forth on those little microtubules. And then even within the cells that aren't so big, a lot of times mitochondria need to be moved from one side of the cell to the other, and those things can grab onto those and walk them from one side of the cell to the other and things like that. So it's a pretty fascinating part of the way that cells work. So I don't think we need to do this question, but which one would it be? Yeah, so those microtubules, yes, C. Good. Okay. All right. So another thing that we find in cells and sticking out of cell membranes are cilia. Okay, and so cilia are these kind of little whip-like extensions. Okay, and they're always in large numbers. So a lot of times it looks just like little hairs on top of the cells. Okay, and the function of these is to move particles across the cell. Okay, so the cilia in the respiratory tract is a great example of this. When you breathe in some air that has some dust in it or some other kind of particle in it, you have this mucous membrane lining our respiratory tract. And so there's a bunch of mucus that some of that dust will stick to. So then it just kind of gets stuck in the dust, but then the cilia that the mucus is around start beating, and they're not just kind of beating randomly, they're beating in the direction that actually pushes that mucus back up, right, and gets back up into your mouth so you can hawk up a loogie, right, and then either spit it out, or you can just swallow it down your digestive tract, which is just as good as getting it out of your body as well. So, so the cilia are actually beating that fluid back upwards. And this is just a short video that kind of shows this process. It'll take 1 sec to load. So you can see those little cilia beating there. You can actually see that there's a little particle there. Not yet. It's starting to come in your view on the top of the screen there. That upwards. So the cilia are definitely microscopic, right? The little particle is microscopic for sure. Seeing it under a microscope and seeing how it can move those little things. So that's pretty much how that works. They always pick the coolest music to go in those videos. Okay. Where are we at? Okay. All right. So then really we're just kind of talking about different parts of the cell, including the cell membrane, and then the things that stick out of cells, like the cellular extensions. So 2 other types of cellular extensions are flagella and microvilli. Okay, flagella, most commonly seen in sperm cells. Okay, so it's just one, it's kind of like a cilia, but it's larger and it's just one and much more powerful. Okay, and the function there is to move those cells. Okay, and then the microvilli are even smaller than the cilia, very, very small little hair-like projections. And we find these most commonly in the digestive tract, especially in the small intestines. We'll get into that next semester in the digestive system. But these are going to be covered with little digestive enzymes that are called our brush border enzymes, which are there to break down carbohydrates, proteins, lipids, to help us break those down and to be able to absorb them in our digestive tract. Okay. All right, so this set of notes just kind of hits on a bunch of different topics of cellular physiology, so it feels like it bounces around a little bit, but we're going to transition and talk a little bit about diffusion, okay, which is definitely connected to the solution stuff that we were just talking about. So the definition of diffusion is this tendency of molecules or ions to move from an area of higher concentration to lower. Okay, and there's nothing really like magical about this, that like somehow the lower concentration is like pulling the higher concentration towards it, anything like that. It's really just the fact that all of the molecules and ions are moving randomly, right? They're all just kind of moving around randomly. And since there's a bunch of them in one place and not that many in another place, the random movement will eventually mean that some of those ones that are in the area of higher concentration will end up in the area of lower concentration. Okay, and when we're talking about diffusion across the cell membrane, it happens really fast because that's very, very thin. A cell membrane is about 1/1000th of the thickness of the piece of paper that you have in front of you. So obviously extremely thin. So very small distance that these have to travel. Okay, so we're going to talk about some different types of diffusion here. And so the most, the simplest form is called simple diffusion. So simple diffusion is substances that can just pass right through the cell membrane. Okay, so substances that can pass right through it. So they have to be non-polar, lipid-soluble substances. Okay, so we have some vitamins that can do that, but then primarily oxygen and CO2 can do that. We don't need any special carriers to get oxygen or CO2 through the cell membrane, which is good because we want those things to move rapidly. Okay, so that's simple diffusion. Facilitated diffusion, there's a couple different types. There's carrier-mediated, where a protein literally kind of grabs onto something, kind of moves it across the cell membrane. Or there's channel-mediated, where there's just an open channel that only allows a certain type of molecule to move through. Okay, and all 3 of those are diffusion that things are moving with their concentration gradient. Okay, so it doesn't require any energy input for that, because the energy input is just the fact that diffusion is going to push it from higher concentration to lower. Okay, so those are three types of diffusion. But then when we talk about moving something against its concentration gradient, it's a whole different story. We need energy to do that. Okay, so we want to take something from a lower concentration and put more of it in where you already have a higher concentration, then that's called active transport. Okay, so movement of molecules against their concentration gradient. And there's two types in the body. Primary active transport means that we're using ATP, which is the chemical form of energy in the body, to power some kind of a protein pump that grabs onto something and slings it across the membrane. Okay? So that's primary active transport. And then secondary active transport is that when we have the concentration gradient of one molecule or ion that wants to push it in a certain direction, we can kind of harness the energy of that to also grab onto something and push that one against its concentration gradient. So that one I kind of think of like a catapult, right? Some of those movies you watched, maybe Game of Thrones or something like that, where there's always those period pieces where they show the big catapults, right? They got it looks like a teeter-totter, right? And there's like a rock or probably like a fireball on one side, right? And then they drop a big rock on the other side. And so there's a lot of energy in that rock that they're dropping, and it's able to use its energy to fling that other one across, right? And that's really what secondary active transport is. We have a lot of energy in one molecule, so it rushes across the membrane. And when it does that, it also flings across another one that is going to require energy to go in that direction. Okay, so here's what I want you to do for a sec. Just take a look at the slide with the people on your team and just kind of differentiate which one of these images is each of these. Okay? And so the one I want you to send on your clicker is the letter that's representing secondary active transport there, but I want you to label all of them in your notes. Okay? Go ahead, talk about that with the people around you.
This one is active. So wait, I don't know where second string. Oh, what is there? Wow.
Yeah.
Or channel mediated. Because if you think about it, it's a third or the second. And then
I thought it was going to be terrible to end up speaking with everyone.
All right, so you're sending in the one that is secondary active transport on your clickers. Is it open? Yeah, it is. So send some more in. Only got 15 of you so far.
and it's going, this one's going with the concentration gradient, and that's what the smoke.
All right, give you about 10 more seconds. We'll send in that one. Secondary active transport's the one you're sending in. I think some more clickers came in the mail, hopefully. What are we up to? Let's see. 52. Is that it? If you just got your clicker, remember you got to hold down that power button and then press AB for the frequency. Getting close. Getting close. Not quite up to 70 yet, but. All right, everybody got it in? So just, yeah, anybody still working on it? Raise your hand if you still haven't put it in. Give me an idea of how many people have their clickers. Okay. All right, so let's take a look and see what you said. Okay, so we're a little spread out there. Normal distribution almost. Not what we're going for typically, but that's okay. That's all right. But we are really between C and D. Okay, so it tells us something about C and D. Okay, C and D. What does C and D have in common? They both what? They both have proteins involved for sure. What else? What about concentration gradient wise? Are the things moving with their concentration gradient or against? Yeah, for the most part against, right? Except for the sodium in there. Okay. Sorry, let me reorient myself on how to use the laser pointer here. Here we go. Okay. Yeah, so here we've got glucose going from the outside of the cell where there's only 2 of them to the inside of the cell where there's a lot of them. So that one means we're definitely going against the concentration gradient there, but the sodium is going with its concentration gradient. Okay. So sodium is like the big rock that they drop on the other side of the catapult. Glucose is sitting down in the catapult. And when the sodium hits it, it flings the glucose across. All right. So which one is that? Secondary active transport or primary active transport? Secondary, we got it. Most of you said it. Secondary active transport, okay? We're using the energy in a concentration gradient of something else to move another one against its concentration gradient, okay? This one here is a very prominent protein in all of our electrically active cells called the sodium potassium pump. It uses ATP to push 3 sodiums out of the cell and 2 potassiums into the cell. Talk about that more when we get to the nervous system again. Okay, so we got that. So that's primary active transport. What about A? What's A? Channel mediated. Okay, we got just an open channel there that lets it go through with its concentration gradient. What's B? Simple diffusion. Good. We got things just going straight through the membrane. Okay, water can do that as well. Okay, and then lastly, E. What would this one be? Carrier mediated. Yeah, we have a little bit more of a specialized protein that can grab onto something and then kind of shuttle it across the membrane. Okay, let me get this guy out of the way. All right, any questions on any of those? Okay. All right, so all of this is related also to osmosis, okay? So osmosis is, so again, diffusion is a movement of a substance from higher concentration to lower, okay? But then osmosis is diffusion of a solvent, typically water, through a selectively permeable membrane. Okay, and osmosis is going to be slightly different. It's not so much about how much water there is on either side. It's about the relative level of water versus solute on either side of a membrane. Okay, so a couple different ways that water can pass through a membrane. One is that we have the lipid bilayer. The cell membrane is kind of wiggling around all the time, and that a lot of times opens up a large enough gap for water to squeeze through. Okay, but then secondly, we also have specialized little channels called aquaporins that allow one molecule of water to go through at a time. Okay, so a very important definition here in term is called osmolarity. And so osmolarity is in its most basic form, the concentration of all the solute particles in a solution. Okay, so not the water part, but how much of the other stuff do we have in the solution, right? How much of that solute do we have in a solution? And remember, again, solution is the solute plus the solvent. So osmolarity is specifically looking at how much solute do we have. Yeah, go ahead. That just means concentration. So kind of a short way to say it, concentration. Thank you for asking that. Okay, other questions? Okay, so a little bit more about osmosis. So this idea of osmotic pressure. Osmotic pressure is the tendency of water to move in or out of a space due to osmosis, okay, or due to the osmolarity of a space. Okay, so typically when we think of pressure, if we think about let's say a plastic bag that we fill with water and we put a bunch of pressure on it, that pressure would tend to push the water out of the bag, right? Or we take a apple pouch. That's what I'm thinking of because I'm throwing those at my kids all the time when they say they're hungry. So you squeeze that little apple juice, applesauce pouch, right? And the pressure on it squeezes it out, okay? But that's actually the opposite of this. When we have high osmotic pressure, instead of pushing the fluid away, it actually pulls it towards it, okay? So high osmotic pressure is going to be due to particles, to having more particles relative to the fluid. And low osmotic pressure is going to be the opposite. It's going to be less particles relative to the amount of fluid that you have. And so high osmotic pressure will pull fluid towards it. Okay, so that's the opposite of squeezing the applesauce pouch. All right, it's a different kind of pressure. This is a pressure that pulls instead of pushes. Okay, so low osmotic pressure doesn't have the ability to hold on to fluid, so it kind of lets that fluid go. Okay, so if you've got a solution with high osmotic pressure, what would that mean for its osmolarity? Would it be high or low? Osmolarity is a measure of a concentration of solutes in a solution. So if we got high osmotic pressure, do we have high or low osmolarity? High, absolutely, yep. If we got low osmotic pressure, we have low osmolarity. Okay. All right, so with this example here, walk through this with the people on your team, okay? Read those questions, read what's going on there, and then why don't you just answer that very last question, which is which way will water flow? Left or right?
Come back to our last question. I still don't get that, but all right. Okay. Higher is the lower.
Nope,
Okay.
high goes with high, yep.
High goes with high.
Yep.
Okay.
All right, so you're just sending in A or B for that last question there. Which way will the water move? Will it move to the left or will it move to the right? A or B? So the blue ones are the water, the green ones are the solute. All right, so go ahead and vote if you haven't yet. I'll give you about 5 seconds. Okay, there's about 6 more of you that need to vote. Go ahead and send something in. Remember, you get those participation points just by participating. Just need to answer the questions. All right, we got 62 now. Good. Okay, let's see what you guys said. Okay, so we're not totally split. Can you guys see that? This is always a pain. Oh man, all right, here we go. Yeah, there we go. Yeah, so 80% of you are saying A, which means you're saying that it moves to the left. Okay, so that would be moving from B to A is what you're saying, right? Yeah, so from B to A. Okay, good. That's correct. Yeah. We'll walk through those questions there. Let me just get this thing out of the way again. Okay, so let's walk through those questions there, and then I'll answer any questions you have. So which side has more solute, A or B? A. There's more little green things on the A side, okay? Which side has higher osmotic pressure, A or B? A, because those are going to go along together, right? Solute or osmotic pressure is dictated by solute, okay? Which side is hyperosmotic? And I haven't really given you those terms yet, but hyper is going to mean high, hypo is going to mean low. So hyperosmotic is always going to go along with the side that has higher osmotic pressure. So which side would be hyperosmotic? A. Which side's hypo? B. And that's how these always work. It's always relative. You can't just say something's hyperosmotic if you're not comparing it to anything. Okay, those are relative terms. Okay. So which way will water flow? Water is going to flow towards the side with more osmotic pressure, towards the side with more solute. And so that's going to go to the left. Okay, questions about that? Go ahead. Yep, absolutely. High osmotic pressure goes with higher osmolarity. Low osmotic pressure goes with low osmolarity. High osmotic pressure goes with hyperosmotic. Low osmotic pressure goes with hypoosmotic. Okay, other questions on that? Okay. Okay, so that's what it looks like afterwards. That water moved to the left, just like you guys said. Those of you that flipped the page over saw that before you answered the question. But, okay, so 2 practical examples of what this means for us, okay? How many people have sprained their ankle before? Well, a lot of us, right? Okay, one of the things that happens when you sprain your ankle is you break cells, okay? You actually damage tissue, okay? So there's a cell getting broken. You also break blood vessels. Your ligaments aren't that vascularized probably, but you definitely break some blood vessels, okay? All of those things lead to stuff spilling out into that space that I mentioned before, that interstitial space, okay? That space that is not inside your cells, not inside blood vessels, but it's everywhere else. So if we're all of a sudden adding a bunch of solute to that space, then what happens is we increase the osmotic pressure in that space, right? So some of you that have sprained your ankles know that sometimes the swelling just hangs around for weeks or for months, for a long time sometimes, right? And part of the problem is because you've got a bunch of extra stuff out in the interstitial space that takes a long time for your body to clean that up. And so you just have higher osmotic pressure out there. So no matter, even if you massage it and push all that fluid out, it's going to come back as long as the solute is there, right? Because it's going to pull that water right out of your blood vessels again, right out of your cells adjacent, and it's just going to keep coming back because of osmotic pressure. Okay, so all that stuff gets spilled out. And what can happen to the cells in the area in a hyperosmotic situation is the cells and blood vessels actually kind of lose water, so they kind of shrink down, right? Okay. All right, so are there any questions about that one? We have a little time, so let me throw one more practical example at you. So eating fiber, right? How many of you are conscious of trying to eat a lot of fiber in your diet? Anybody think about it a little bit? Yeah. Yeah, once you get older, you'll start thinking about it for sure. but so fiber is made up of the cell walls of plants, right? Those plant cell walls, and we can't break it down. Our digestive system doesn't have the ability to break it down. So it stays in our digestive tract. Okay. So as we eat, all that stuff that's getting brought into our bloodstream gets brought in. But then if we have a lot of fiber in our diet, then we got a lot of solute left in the middle of our digestive tract passing through. So when that stuff is passing through our digestive tract, it's passing by all the cells of the walls of the digestive tract, as well as the blood vessels around it. And since there's extra solute because of that fiber, then we have higher osmotic pressure in our digestive tract. Right? So if we got higher osmotic pressure in our digestive tract, then that means we pull water out of the cells lining the digestive tract, as well as pull water out of the blood vessels lining the digestive tract. So more water in your stool, the softer it is, the more regular you are. Right? So that's how the high fiber diet leads to more regularity. The extreme version of that would be you get the stomach flu, right? And all of the lining of your intestine gets inflamed to the point where you can't absorb anything, right? So everything you eat is staying in your digestive tract. So you've got a ton of osmotic pressure because you got a ton of solute, and you got to get up and go to the bathroom quick, right? So it's going to run right through you because a lot of water came in, right? So a lot of water means it's going to move through fast. That's how you get diarrhea, right? Okay, thumbs up for diarrhea. I don't know why I did that, but- All right, any questions about that? I don't know why I went right to thumbs up after diarrhea. There was a thumbs down for diarrhea. Thank you. Okay. All right. All right. Okay. Let's do a few examples here with some clicker questions. So this is kind of the classic example of we take a red blood cell and we drop it in a solution. And that solution may be hyper, well, yeah, let me first talk about that term tonic first before we do these questions, okay? So we talked about saying hyperosmotic, hypoosmotic, but the tonic ones, when you see tonic in there, so that comes from a term called tonicity. And so tonicity is referring to a solution's ability to change the shape of a cell. So write that down for sure. Tonicity equals a solution's ability to change the shape of a cell. So anytime we see tonic in one of those words, we won't use that to talk about the inside of a cell. That doesn't make sense. We can only use that to talk about the outside of the cell, the solution that cell is in. So tonicity equals a solution's ability to change the shape of a cell. Luckily, the hypertonic goes right along with hyperosmotic, goes right along with high osmotic pressure. Okay, hypo goes right along with it as well. It's just a little bit different because we can only use it talking about the outside of a cell because it's talking about how we change the shape of that cell. Okay, does that make sense? Questions on that? Okay. All right, so let's do this first one. So which picture below shows a hypertonic solution outside the cell? And again, hypertonic would only be referring to the outside of the cell, but I was just making sure it's crystal clear for you there. All right, so you're just sending in A, B, or C. If you're not sure, make sure to ask the person next to you. Discuss it. All right, couple more people. Go ahead and send it in. We had 62 last time. Anybody else? We lost 5. That's not good. Okay, remember you got to respond to all these to get your participation points for the day. All right. Guilt didn't help. Not sure. Okay. All right. Okay, so most of you are saying B. Okay, so B is the one that's hypertonic. So we see a lot more of that stuff on the outside of the cell than we do on the inside of the cell. Okay, good. Next one. Okay, let's do this one. Which picture below shows a cell in a solution that is isotonic. Just give you about 30 seconds to answer this one. Go ahead. Yeah, we didn't really talk about that one, huh? Yeah, good question. Isoosmotic, yeah. So the iso means the same. Okay, so hypo means less, hyper means more, iso means the same. Okay, so this would be the solution on either side of the cell membrane is pretty much the same. Okay, or if we're talking about tonicity, we would say the solution the cell's in is a similar osmotic pressure to the solution inside the cell. Okay, go ahead and vote if you haven't yet. All right, anybody else? There we go. Okay, see what you guys said here. Okay, so most of you are saying C, right? That's the one that's the normal shape of a red blood cell is like that. And we have about the same amount of solute outside as we do inside. Okay, let's go to the next one. Okay, hypotonic solution outside of the cell. Okay, just yell it out. Hey, got it. Okay, let's go to the next one then. All right, now this one is a little trickier. So go ahead and talk through this one with the people on your team.
Below shows the situation where osmotic pressure
Bring it all in, right? Because the water wants to go out. So it's gonna go towards that, which is it's suck dry. It's like a little eat it. Beef.
All right, so whenever you're ready, send that in. No, not for this one. The previous one we did because it was just kind of the last one left. All right, a couple more people. Go ahead and send it in. Anybody else? All right, it's almost there. Okay, let's see what you guys said. All right, good. So most of us are saying A. Okay, so a situation where the osmotic pressure was high inside the cell, i.e. the osmolarity was higher inside the cell, or we could also say that the cell is in what type of solution if we're talking about tonicity? What would we say? Hypotonic, right? Hypotonic solution. Yeah, I'm just trying to get that little box out of the way. I realize you guys can't see it. The rest of the question, there we go. Okay, yeah, and so that led to osmosis. So most of you said A. So what? So when we put a cell in a hypotonic solution, the risk is actually bursting that cell, called cell lysis, right? If we let too much water get into that cell, it can burst, okay? It has the ability to expand quite a bit, but if it tries to expand too much, then it bursts, right? So that'd be cell lysis. Okay, one more here. Okay. Talk through that one with your team and vote when you're ready.
Be old and know a lot. I need to take my classes.
That's why you're here.
Right. It was so funny. I just had
No way.
I went to the nurse and she's like, you should use crutches. I'm like, I'm not using crutches.
Look, no.
On the bus every day. Like,
All right. Send it in if you haven't yet. Go ahead and send it in. Couple more people. Give you just about 5 seconds to do that. All right, anybody else? Okay. See what you guys said. So which picture below shows a situation where the solution outside the cell had a high osmotic pressure, i.e. osmolarity outside was high, and that led to osmosis. And so you guys said, everybody said B. Fantastic. Good job. That's a wonderful way to end the first week of college for you freshmen and first week of class for you not freshmen. All right, guys, have a great week. Great to be with you. Great weekend, sorry. And we'll see you on Wednesday. Okay, Wednesday. Have a good Labor Day.
I get off work on campus like between 1 and 1:15, so there's a few days that I might